IB Bio Cell Biology Part 3 will focus on prokaryotic cells, cells that lack a nuclear envelope surrounding the DNA. Prokaryotes have a simple cell structure without compartmentalization. The essential idea is that eukaryotes have a much more complex cell structure than prokaryotes. Here's an outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the unit that you need to review. This movie is focused right here. Scientists have grouped all of the world's organisms into 3 domains, here in purple: the archaeans, the eubacteria, and the eukarya. The archaeans and the eubacteria are prokaryotic. Their cells lack compartmentalization. They lack a nuclear envelope around the DNA. The eukarya domain include all organisms whose cells have a nucleus, including the protists, fungi, plants, and animals. It's important to note that viruses are non-living biological particles. Here's a diagram displaying the 3 domains: archaeans, eubacteria, and eukarya. The common ancestor is here. Archaeans are prokaryotic cells that tend to live in extreme environments, hot or salty. Archaeans have certain characteristics that suggest that they're more closely related to eukarya than eubacteria are to eukarya. Prokaryotic cells, the archaeans and eubacteria, lack compartmentalization, as can be seen in this micrograph. There is little internal structure. Prokaryotic cells have various characteristics. They tend to be unicellular, small cells, 1 micrometer, no nuclear membrane. There is no membrane around the DNA, few internal organelles. There is a cell wall composed of peptidoglycan, not cellulose. There can be flagella, but they're specific to the bacterial group. A single loop of DNA not associated with protein. Small ribosomes. They divide by binary fission, and they have varied nutrition. Here are images of prokaryotic cells. It's important to remember how small these cells are. Thus, without electron microscopes, we would not easily see any internal structure, as you can see here in these light microscope images. In this micrograph, we can see a single bacterial cell with its single loop of DNA extruded from the cell. Prokaryotic DNA is not associated with histone protein, and in addition to the large circular loop, there can be small rings of DNA known as plasmids. Could you calculate the magnification of this image using the given actual size here? Bacterial cells have varied metabolism. Some are photosynthetic, and some fix nitrogen from the atmosphere. And some cells function without oxygen. They're anaerobic. Here is another micrograph of a bacterium dividing into 2 by a process known as binary fission. This is how prokaryotes divide. Notice the lack of compartmentalization in these cells. Notice that there's a region called the nucleoid region where there's a single loop of DNA, the 1 chromosome of DNA, but it's not bound by a membrane, and the DNA is not associated with histone protein. Here's a drawing of a prokaryotic cell. Take note of the structures that I've emphasized in red lettering. The nucleoid region is here, where there's a single loop of DNA that lies within the cytoplasm of the cell, free of histone proteins. All cells everywhere have a cell membrane, a plasma membrane. There are small rings of DNA known as plasmids. A bacterial flagellum is a unique structure, but serves to provide motion to the cell. The cytoplasm has many small ribosomes, enzymes that catalyze protein synthesis. The 70S ribosomes are slightly smaller than the ribosomes found in eukaryotic cells. The pili serve to adhere the bacterium to a surface, and the cell wall is composed of a substance known as peptidoglycan. Here is another drawing displaying the same structures introduced in the last slide. Keep in mind that the cell wall protects the cell. The plasma membrane regulates what goes in and goes out of the cell. And the cytoplasm is a solution within which chemical reactions occur. Here's another image. What is the name of the region where the single loop of DNA lies in the cytoplasm? How large are the ribosomes? And what is the function of the pili? The cell wall serves to protect the cell. The cell membrane regulates what goes in and goes out. And the cytoplasm is a solution within which chemical reactions occur. Bacterial cells often have a slime capsule around them. The cell wall here is composed of peptidoglycan, a protein-carbohydrate complex. Pili serve to adhere the cell to a surface. The important IB syllabus statement for this movie is right here. Draw and label a diagram of the ultrastructure of Escherichia coli, which includes the following structures: cell wall, plasma membrane, cytoplasm, pili, flagellum, 7DS ribosomes, and the nucleoid, which is the region containing the naked DNA. State one function for each structure. Here is a micrograph of a prokaryotic cell, Escherichia coli. This bacterium lives in your gut, happily so, as you provide it with a steady stream of food and a warm place to live. E. coli bacterium, like all bacteria, have very little compartmentalization beneath the cell membrane, and they have 7DS ribosomes, which serve to enzymatically catalyze protein synthesis. Here's another IB syllabus statement. State that prokaryotic cells divide by binary fission. Here's a micrograph of a bacterial cell undergoing binary fission. As it splits into two, at this point the other structures should be familiar to you. One last chance to see an image of a prokaryotic cell. In this slide, I have once again emphasized the unique characteristics of prokaryotic DNA. There's a nucleoid region, there's no nuclear membrane with one circular chromosome of DNA. The DNA is not associated with protein, not associated with histone protein, and prokaryotes have plasmids, these rings of DNA that lie separate from the single loop in the nucleoid region. Here's a micrograph showing the plasmid of a prokaryotic cell. Plasmids are small loops of DNA separate from the larger single loop of bacterial DNA. Plasmids carry important genes, such as resistance to antibiotics, and are now used as tools to transfer genes between species in gene technology. Stay tuned. Here is a prokaryotic cell that has just finished undergoing binary fission. You can study the structural details. Prokaryotic cells do have flagella to provide movement. However, bacterial flagella are not at all the same structure as eukaryotic flagella. Whereas eukaryotic flagella are completely surrounded by the plasma membrane and they move side to side, bacterial flagella are an external structure poking through the membrane. As well, bacterial flagella rotate in a spiral motion. So here's the IB syllabus statement that you need to gain control of. Compare with detail at least 5 differences between a prokaryotic and a eukaryotic cell. Be sure to include 2 differences between nuclear material. State at least 2 similarities between the 2 cell types. Not having studied eukaryotic cells yet, you may feel a bit uncomfortable with the comparison, but eukaryotes is the subject of Cell Biology, Part 4. Here is a slide that provides the comparison between prokaryotic cells on the left and eukaryotic cells on the right. I'll let you study this slide on your own. You may want to return to this slide after you have studied eukaryotic cells because you need to get control of the differences between the 2 cell types. And that brings us to the end of Cell Biology, Part 3. Eukaryotic cells are next.